Oh, all right. Um, yeah, really, thanks a lot for giving opportunity to give this lecture to the student in Korea University. As the Professor Lee introduced me, I'm Yura, currently staying in Brussels in the university, like Free University of Brussels, if I translate in English. And our lab is, as you can see on the name, we are mainly studying about the stem cells and cancer. So today I will give a lecture about how the innate immunity and FKB pathway is controlling prostate stem cell and how the cells are getting plasticity and how it's connected to the tumor initiation. So before starting that, I have a disclaimer that all the content that I will show today is included in this publication. And in a September issue of Nature Cancer, we have a cover image. And I will definitely show this QR code later on. So we have a publication and the research briefing, like a short introduction of our paper, and the GitHub repository, which included all the codes and algorithm used in this analysis. Uh, actually, this project cannot be done alone. So this is my supervisor and this is my co-supervisor. And definitely we have a lot of collaborators who put effort in this project. And the one who is working for administrative work in our lab, definitely without her, we cannot do any work in the paperwork, let's say. So we had these colleagues who made this work together. Before I start about the story that I have to tell how the stem cell potency is defined. So there are actually four different stem cell potency. The first one is called totipotent. So we have a one cell, and definitely this cell can make the full organism. Like for example, the best example is that we have a one egg and one sperm, and they make exactly same like the homogeneous twins. So if we move to the a bit more and more and more specific, that the next one is a pluripotent. Definitely the pluripotent start from the one cells. And in the end, if it goes further and further, it makes whole fetus. So one cell doesn't have any limitation of the functions or making the specific cell. It can make everything. However, we have another stem cell hierarchy called multipotent. So it's similar to this pluripotent I showed, but one of the different thing is that the cells which are made after this multipotent stem cell is limited function. For example, like here we have multipotent hematopoietic stem cells. They will make the different cell types, but those are all related either immune or the component of blood. And the very last one is unipotent. As you can see, the name uni means that it only makes one specific cell type. The one of the good example is that how the sperms are made. So here that the one undifferentiated spermatogonia starts, and they started doing more and more differentiation, and in the end they make the one type of cell which is called sperm. So definitely there are four different types of potency, and then let's go a bit more specific for the cell. So we start from stem cells, and this stem cell can do the self-renewal, which means that stem cells are making same stem cells. Not only that, it can go to the differentiation, which means that they are started making the specific type of cells, and in the middle we say transit amplifying cells. So these transit amplifying cells can either differentiate further or either still do the self-renewal to keep this population. And in the end, we see the differentiated cells, which one already has a specific function, and it will just keep as it is, it's unipotent afterwards. So one of the technique that we use in the lab quite frequently is called lineage tracing, because we already told on the previous slide that the stem cell is going to transit amplifying stages and it becomes a differentiated cells. The question is, what kind of these differentiated cells are coming from? So we use a specific model of a mouse composed of different cassettes. And then one of the most important thing is called YFP or GFP, which is kind of giving fluorescent light to the cells, which is permanent, and it can be transit further. So if we treat tamoxifen, for example, to activate this CRE-ER promoter, this lock site between the stop codon will be closed, and the YFP can express in the cell. So if we are tracing the multipotent stem cell, for example, it will still this YFP signal will be a transit to the TA cells, and then you can see the differentiated cells. So we can say all these differentiated cells are coming from one stem cell. However, if we are tracing the unipotent stem cells, they will mark only one and the E will transit only one, but the other cell types will never have YFP. So we can conclude all those cells are unipotent. And definitely the next question is that we have a stem cells and progenitors and how we can compare and how we know which is progenitor in this lineage tracing technique. The easiest way is that put the YFP in the middle, like we not marking the stem cells, but using the different promoter in the mouse model, we can mark the progenitor specifically. And afterwards, if this progenitor express YFP or GFP, the reporter, they will go further on the specific cell types, and we can say, oh, this progenitor is making C and D differentiated cells. So since this study is mainly talking about prostate, so we need to understand how the prostate looks like and how the cell types are composed of. So most of the people know that the prostate is under the bladder, and then in the between the urethra is passing by. However, it's for human. On mouse prostate, it looks like this. So it's actually between bladder and urethra. And also it's composed of like ventrolateral, dorsal, anterior, different lobes, and then seminal vesicle where they make semen, and ductal deferens, which is connected to the other organs. So actually, if we are cutting the prostate in anywhere, like horizontally, we can see this sort of structure, which is composed of the basal cells, luminal cells, intermediate cells, which means like a basal, between basal and luminal, let's say, and then which is a rare population called neuroendocrine cells. And the 4 different cell types can be marked by different markers, as you can see, and these markers are mainly used in pathologists. So definitely the question is how we understood this multipotent basal progenitors contribute to the luminal and the neuroendocrine cells. Actually, there's a publication from our lab in 2012. So we use this model, still we use a YFP model. And this one, the K14 is actually the basal marker genes. They will trace the cells from basal cells. So and 3 days after birth, as you can see that the basal cells are only the basal cells, and as well on using the other markers. However, after 4 weeks, which we count after puberty of a mouse, all the Ka cells are having YFP positive, which means that those luminal cells are coming from basal cells on here. And also the synaptocystine, which is marking the rare neuroendocrine cells, as well as you can see that there they have a co-expression of YFP, which also indicates that those basal cells can make specific cell type, not only luminal cells. So after several years after one of my old colleague was doing to understand whether this multipotency changed unipotency, and is it region specific or not. So she tried to track the cells after birth, during the development, and the puberty adult, which is after 21 days of birth. So at the very beginning of the birth, regardless of where cells are located, all the basal cells are multipotent. However, it started to developing. We see that the tip, which is the end of the prostate branch, it's still multipotent. However, the duct, which is on this the branch look like they are unipotent. So it means that tips still can make luminal cells, but duct never make the luminal cells anymore. And after postnatal 21 days, we see that regardless of where the basal we are tracing from, all of them are unipotent. And this is defining the 2 different studies. So here, if I mention again, I mean, it's not only ROSA 26, but we can use different cell types. And one of the study in our lab in 2019 as well, tried to understand how this cellular hierarchy works. And so we use YFP still, and we define that there is a K14 and K5 basal cells and luminal cells, and still these basal cells are giving rise to luminal cells. So still we say the cellular hierarchy is always basal to luminal in prostate gland. So I already explained that the multipotent cells are changing to unipotent after the puberty. And actually, there are several approaches to understand how this multipotence is coming from and how it can be reinduced after the cells are unipotent. So we already know that in embryonic status, it's always multipotent, and regardless of cell types, basal, luminal, whatever they are, they can make each other. And the one thing they try, it's actually like really old approach. Like this was done like in 1960s, and they transplant only the basal cells of a prostate, it can form the full structure of a prostate, which means that the basal can give rise to luminal and the neuroendocrine cells. And the one of the study which was done in the other team in 2012, that if they are ablating the lineage, let's say like they intentionally kill the luminal cells in the middle, after the cells are unipotent, and still these basal cells can fill the gap, which doesn't have luminal anymore. And also that there are several approaches to see after the auto status if they activate oncogene expression, like a P10, for example. This can give rise to luminal cells from basal cells, and it can go further to the tumor. So those kind of things are already, those kind of research are well known. So the people are started asking them how these are related to tumor for sure. So since I keep talking about prostate cancer, I have to talk about a little bit about the prostate cancer before we're moving on. So as you can see that the normal prostate in a human, let's say in a human, that the prostate gland is right under the bladder and it looks like this. But when there is a cancer started, the tumor started inside of the prostate, and then we can see that those are started being flattened, like it started getting swollen. So we can detect that it's the cancer started. And also, one thing is that the cancer cells are getting into the lumen. So from the basals, it goes luminal, and that it started occupying inside of the prostate lumen. And cell-free DNA or other secretive proteins can go through urethra and with the urine. And why we say the PTEN gene is oncogene-related prostate cancer is that, as you can see that this catalog, prostate cancer has a quite high ratio of a deletion of PTEN, which is called deep deletion. And one of the thing important is that PTEN is actually an oncogene, like a tumor suppressor gene. So as you can see on this this kind of graphics, that the PTEN is usually blocking the PI3K AKT-mTOR pathway, which is promoting the proliferation, cell survival, or angiogenesis. So if the PTEN loss happens, that this will activate all these pathways, which means that they will push forward the cell proliferation with angiogenesis. And this is already well known that it's related to tumorigenesis. And not only with it, I didn't put it on here, but already on the Dana Pierce lab in America, they found out that the PTEN loss is also activated the J and K, like JAK and STEP pathway, which is independent of those pathways. So yeah, I mean, this is just some of the what I explained on the further. So definitely, if you have a PTEN loss, they started having a aggressive form of a prostate tumor, but it's not directly jumping to aggressive form. So like they started having a small lesion of a tumor, and it started being adenocarcinoma, and it started getting further, and sometimes it goes metastasis, and the PTEN deletion is significantly contributing to those steps. So if I show that the result of our paper, that the PTEN deletion is actually selective. So if we are targeting basal cells, first of all, and we do the PTEN deletion, what happens is, as you can see on this facts chart, the first week of induction, we only see the basal cells, the YFP positive, but we start to see more and more luminal cells. And you can see that this is actually quantification of those plot. And what we observed was that, especially on the anterodorsal prostate, we see that there is a YFP marked luminal cells are significantly increasing by the time passes after induction. Also, the one thing we observed after the PTEN deletion on basal cell is that we start to see that there is a hybrid region, which is not only marked with K14, but as well as K8 and the YFP together. So which means that after the PTEN deletion on basal cells, it gained the multipotency, and it can make the luminal cells again, even though the mouse itself is a unipotent status. And also, when we target the PTEN deletion on basal cells, what we observe is that from the 6 weeks, let's say, we started observing the different tumor types are occurring. And as you can see, on the anterodorsal status compared to ventral, they have a more invasive form after the time goes further. However, when we target the luminal cells, we totally see different thing. It doesn't have any multipotence, as you can see. Even though we wait 12 weeks after deletion in luminal cells, we don't observe any basal cells, and also which is marked as a YFP basal cells. And also on this, when we do the staining, we see that there is no co-expression of keratin 14 and Ka, which are marker of basal luminal cells, as you can see here. So which indicates that after PTEN deletion, this plasticity is only happening in basal cells. And even though we do the staining of the tumor, as you can see, we don't see any differences, and it's way, way slower because we already observed the tumor is happening on 6th week on basal, but some cases, it's really like a light form, or it doesn't go that much invasive, even though we wait super long time. And then one of the study actively studied is that what if there is a co-mutation? So which means that not only consider PTEN mutation, we combine with other mutations such as a PIK3CA mutation, which is well known in the breast tumor. Well, in that case, what we observed is that their plasticity is way faster, because you see that if we only induce a PTEN deletion in basal cells, you can see their luminal cells are like less than 5%. However, when we activate the PIK3CA mutation together, as you can see that it's already even over 10% in the certain lesion. And also the compared to VP, the DLP APs are way faster. And that when we do the staining, we see the exactly same phenomena. And one of the other mutations, the P53 mutation. So if we combine PTEN and PP3, we see exactly same pattern as we observe in the PIK3CA mutation, as well as they have a co-expression of Ka keratin 14 YFP on DLP and AP, but we didn't observe it in the ventral prostate, which is farther than DLP and AP. So it's also not only that they are faster, but the co-mutation, the plasticity induced by co-mutation is regional specific. So yeah, definitely we do the same thing. So what if we treat the adenovirus to do the same approach, not only just waiting for the induction, then definitely we observe the same phenomena, and we have really high ratio of the luminal cells compared to the hybrid of beta luminal, which is marked as YFP. So which means that compared to the hybrid, it's already shifting to luminal cells after the PTEN induction. So the one thing I already told on the introduction is that the mTOR pathway is blocked by a PTEN. So what if we have a PTEN deletion, and then definitely mTOR pathway will be activated. And what if we inhibit mTOR mTOR, which is really the end of the axis directly, whether it will block or not? The answer is actually yes, because we see that it's actually after 6 weeks of deletion on the basal, and we still see that there are hybrid cells, as you can see here. But if we pharmacologically inhibit the mTOR axis, then we don't see any co-expression anymore. And as well, yeah, it's the other example. Like if we combine PIK3CA and PTEN together, we also observe the exactly same phenomena, and that we don't see that much of a luminal cells marked YFP compared to we just activate those mutations. And also in the cancer status, we see totally different after blocking the mTOR. So we see that on the PTEN deleted status, we can already see that there are quite several different tumors. But in a lapamycin treated, which is blocking mTOR pathway, we see that the clear structure of the prostate, which has a luminal and basal status. And when we are quantifying that the phenotype incidence, as you see that after the rapamycin treatment, we only see the normal cells, no more tumor cells anymore. So definitely blocking the end of PI3K, AKT, mTOR pathway, it's already held to block the plasticity and making the tumor initiation. Then definitely the next question will be then, how does PTEN deletion induce the plasticity, and what kind of mechanism is related? So we decided to do the single cell because We already know that there are several different cell types are mixed. So we really wanted to see cell type by cell type. So the first thing we tried was that we do the control data, which one is a CD1 mice we call, and then after 6 weeks of deletion. So in the control data, you can see that we have a two basal cell type, which is a low expression of P63 and the high expression of NKX3.1. And the four different prostate lobes are in a separate way. However, after the PTEN deletion, we see that there is one another cell type of the basal, which has a high P63 expression. And then there is two expected pathways, which is passing by Hillock cells, hybrid basal, and then going to proximal, which is corresponding to AP I show on the staining. And then also there's another one starting from basal NK3.1 positive and going to hybrid, and in the end goes to anterodorsal ventral. And also when we are quantifying that the marker gene expression of the basal and luminal, which is calculated on this control data, we see that two different hybrid populations are expressing the basal and luminal markers equivalently. And here is just kind of showing some marker that how we define. And definitely here we say that there is Hillock cell hybrid cells expressing K13, K4. The K4 is actually expressing only on proximal markers, and here K13 we don't see anything. And AQP3 is specific on Hillock, some part of Hillock cells near the hybrid junction, and NKX3.1 is on this population way higher. Actually, here the K13 is already known marker of Hillock cells, which is discussed in 2018 and 2019 already. So actually, this Hillock cell is known on airway, like a bronchia or lung cell, the cells from lung or bronchia. However, this team was doing a single cell. Again, they show that there is Hillock cells, which is marked keratin 13, and which is also in the same layer of the basal, but they are not stratified cells. So we found that this keratin 13 positive cells in our PTEN deleted data, so we decided to use this as one of the signature later on. And the next question is that then how these cells are having plasticity and how the direction looks like. So we did the pseudo time analysis. So actually, we found the two pseudo time trajectory in our data. So the first one is actually starting from basal and then going to Hillock, hybrid, and proximal. As you can see there, the proportional marker genes are decreasing, increasing by the pseudo time. And when we do the experiment to see whether those markers, which is changing their gene expression significantly in the pseudo time, whether it makes sense, the answer is yes, because we can see that after, like a control doesn't have a K14, keratin 4 co-expression, but we start to see that the keratin 14, keratin 4 YFP positive in the PTEN, PTEN peak, and P53 PTEN together. So which means that the genes, definitely this pseudo time and the genes significantly changing their expression in the pseudo time, they are related and they can be used as they are related to hybrid status and changing their cell fate. This is also another example that we tested to see where this reprogram happens in the real mouse. And then we see that already in the K13 or AQP3, which are the markers of the Hillock and hybrid genes, as you get hybrid population, you see that AP and the anteroprostate, dorsolateral prostate, they have the co-expression of those cells. However, the ventral prostate, which is farther, which is marked by NKX3.1, didn't show this phenomena. So which means that this pseudo time trajectory is not related to NKX3.1 positive, and what we observe in our data is exactly biologically true. And the another one, as I said, was that the one is related to NKX3.1, which is passing by the hybrid anterodorsal, and in the end it goes to ventral. And NK3.1 expression I already showed was in the contrary, it has a small portion of basal has it, but here the more higher in the hybrid. And definitely here, yeah, we see the bunch of YFP expression. And then when we remove YFP, we can see that on the ventral prostate in this tip part, we see that the expression of NK3.1. And definitely we see that on these cases, it goes to the ventral prostate in the end. We observe that NK3.1 and the YFP K14 are all co-expressed in the ventral prostate way higher. However, we didn't observe that higher ratio in anteroprostate or dorsoventral, and then the quantification is as like this. So then the next question will be like how we can characterize the hybrid basal luminal cells and how this linear trajectory can be explained. So we decided to do the gene differential gene expression analysis to see what kind of genes are activated. So the first goal is understand what kind of genes can be counted as signature compared to basal or luminal cells. And these are the results, and we keep observing like AQP3, which was AQP3, and the K13, which is marking our hybrid, as well as the LY60, keratin 6A. Those kind of genes are already well known in the multipotent business, mainly in mammary gland though, but the mammary gland and prostate are having the same structure. That's why we say this might be also important in the prostate. Not only that, the clue keratin 4, WFDC2, when we compare with the basal cells, which are the marker of the proximal prostate, which is defined by 3 different studies previously. And when we compare with the proximal luminal cells, that we observe some of the marker genes of the basal cells. So we can say that definitely our DG analysis was going well, and also we found some markers which is already known as related to the plasticity. And when we are considering this small population of hybrid marking by NK3.1, they are totally showing different status. For sure, we define the marker genes of those anterodorsal like FGL1, TGM4, NK3.1, where when we compare with the ventral population, we see that the keratin 14, keratin 5, which is a marker of basal. But here we observe that the highest is actually C1RB and RAN1, which are studied already in the prostate, that they can be related to the tumor occurrence. So until with this 6 week of deletion that we can like kind of making the model that from basal in the APDLP, he started making hybrid state, the Hillock status, hybrid state, and then go to proximal like, which is marked keratin 4, keratin 8. But in the ventral status, it directly goes to distal like, which is marked K8 positive and NK 3.1 positive. So depending on a region or like a lesion, they are making a different phenomena of cell fate in infidelity. And the next, okay, so we know what kind of genes are activated and how this trajectory can be composed. However, the next question is that which transcript factor or which regulator can be related. So here, since we only have a single cell RNA-6, so we use the tool called Cynic, which can predict that the transcript factor and their target genes, which are activated in different clusters in single cell RNA-seq data. So in our case, actually, we found quite a lot of immune-related, like interferon 6, 7, and 9, which are activated in the proximal and the hybrid status, as you can see. And as already known, the JAK pathway, we also found on the STAT1 and STAT2, and as well as NF-kappa-B related pathways. So what we tried to do was do the bulk RNA-seq afterward to see whether those kind of transcript factors are really activated and those genes are highly expressed after PTEN deletion. The answer is actually yes, because we see that compared to the basal or luminal control, we see that they are high expression of those inflammation-related marker genes in PTEN deleted both basal and luminal, and as well as those ones that the genes known as reprogramming in the prostate status, and then the genes related to cell proliferation are super high in the PTEN deleted. And cell proliferation is already way higher in the luminal cells, which is a PTEN deletion. And when we are collecting all the genes, which is up in a PTEN deleted cells of the basal and luminal compared to control, we did the gene ontology and pathway analysis. We see that on the basal and luminal, they have innate immunity, inflammatory responses, and neutrophil chemotaxis, like which is really related to TNF pathway and immunity related. But in a luminal case, not only those ones, we also observe the mitosis cell cycle. Definitely, we see those genes. And angiogenesis, which is well known when the PI3K, AKT, mTOR pathway is still activated. And we did an autopsy to see whether what we found in the cynic is true. The answer is yes, because when the peaks are up in the chromatin regions are more open in P10 deleted basal compared to control. Definitely those APO families are always coming up when the cells are stressed, and P63 since we are working on basal, and then NF-kappa B. And when we do the Western blot, we see that the STAT1, P65, and AKT are more activated in P10 deleted cells. And when we check the neutrophil infiltration, we observe that there is more neutrophil infiltration after P10 deletion compared to control. So from this toxic, one of the reviewer was asking that then this phenomena is also region specific or not. So we decided to do the ataxic after the P10 deletion, but only in a basal cells of different lesion. And the answer is, as you can see again, yes, because we see that those kind of peaks after P10 deletion in basal, they are way highly open in AP and DLP compared to VP. And those are having binding site of not only NF-kappa B, but we also find KLF5 which is related to luminal and which is related to as well as the stem cell regulation. So we see that this is again specifically happening in the prostate. So we already said that if we block the mTOR pathway, the mTOR axis, that we don't see any plasticity anymore. Then the question is whether this innate immunity activation is related to mTOR or not. The answer is it's dependent, because when we try to see that after the rapamycin treatment, we can see that their inflammation and the reprogramming cell proliferations are all downregulated. And also we observed that there is no more neutrophil infiltration, and also after rapamycin treatment, we don't see the tumor anymore. So the mTOR when we inhibit mTOR, the innate immunity activation in the P10 deleted cells are not happening anymore. So okay, so we understood like early time phase after the P10 deletion is related to tumor and then related to the plasticity. And then definitely we want to know is that when the tumor happens, like early stage of tumor happens, what is the differences? So here that we induced the P10 deletion for 10 months in mouse, and then we selected YFP positive cells, and then we did a single cell again. So here what we observe is that the cell composition is totally different from 6 months, because we still see that the interferon and all the upper part, which the luminal cells are all positive to the proximal markers, and we still observe basal, hillock, and hybrid, but we don't observe the distal luminal cells, which was marking the ventral and anterodorsal prostate. And also, one of the thing I have to point out is that here a lot of clusters are having expression of chemokine, interferon, MHC class 2 antigen. So we are again confirmed that this innate immunity or inflammatory related phenotype and the gene expressions are persisted until the tumor is happening. So here the regional reprogramming we are answering again. After the 10 months, we exactly see the same phenomena for sure. So here we do the exactly same approach, but after 10 months, like this kind of things happening. And we see that here is way clear in the regional, because we see that those hybrid related, like driving the stem cell plasticity, actually they are way higher in the AP compared to the other lesions. And as you can see in the VP, regardless of which marker we are using, they are way lower. And also we do the same test on the co-mutation of P53 or P10 peak. Definitely we see the exactly same thing. But the difference is that their markers expressions are way clear, and their co-expression patterns are way clear compared to the 6 month already, and their ratio is way higher. So we did a cynic analysis to see whether still this transfer factor we observe in the 6 week of deletion are still observed or not. The answer is yes, because it started having a more clear, but the difference is that those interferon stat and NF-kappa B, they are more observed in the luminal cells, which is already differentiated from basal cells. not in the hybrid status anymore. So as a one of the questions is that can we find the human relevance of our findings, and our marker genes we observed are still persist in human data. So what we try to do first thing is taking the 4 different human data sets which is already published. So one is a ERG positive tumor, another one is actually collecting like 14 different patients, and this one is cribriform, and the other one is having the androgen receptor high and low cells. And then we observe actually that when we calculate the enrichment score of our marker genes, that we define the marker genes of a hybrid and HELOC or proximal status, what we observe is that they are super highly enriched in the club cells, HELOC cells, like club HELOC, those kind of intermediate status regardless of the tumor type. And when we try to see that the specific expression, we also observe exactly the same thing, but their gene expressions are way more clear than enrichment score in general. And also, if you're using those data sets to see whether the pseudotime trajectory is exactly same or not, the answer is yes, because in this case, ERG positive, ERG negative data, we observe that those pseudotimes are either passing by club and then go to the ERG transcript factor positive tumor or going to the luminal. And also the other case, which is having an androgen receptor related tumor, that we see that it goes to anyways 3 different cancer cells in the end, even just passing by androgen receptor high or low. So we can say that what we observe in our mouse model is definitely human relevant. So after this, we collaborated with a pathologist in our hospital, and then we observed that the markers we define in this infidelity, we said that actually those stainings are way higher in the high grade of prostate cancer, which is a Gleason grade of 4 or 5 compared to the 1. And when we also quantify the number of samples to see whether those markers are positive and negative, as you can see that especially PIGR and the STAT1, we see that their positive ratio is gradually increased by the Gleason grade. And AQP3 is also as well when we consider, and there are the number of patients which has a higher, the ratio of the patient which has a high signaling is way also getting higher when the Gleason grade is getting worse. So then definitely we saw the NF-kappa B, JAK-STAT, and the immune-related pathways. And the next question is that what if we inhibit these pathways by pharmacological inhibition? So we use JSH23, which is blocking NF-kappa B pathway, and the ruxolitinib, which is blocking the JAK-STAT pathway. And we treated on both K5P10 and K5P10 mouse. And definitely what we observe is that after inhibiting the NF-kappa B or JAK, what we observe is compared to control, their plasticity is definitely decreased, which means that the basal cells cannot make the luminal cells as the control is doing. And when we stain the data after treating inhibitors, 2 different inhibitors, definitely we see the same thing, because in a control, we see that the co-expression of K8, YFB, YFB, K13, and YFK4. But here, after the treating the 2 different inhibitors, we don't see any co-expression anymore. And this is a quantification of our staining, and we can see again the inhibition dramatically inhibit the stem cell plasticity. And then the other one we test is actually IL-1 by anakinra. As you see that here again, the plasticity after deletion happens in a control, but when we treat the IL-1 inhibitor as well, we see the exactly same phenomena, like not making that much of a stem cell plasticity anymore in prostate. And not only with that, we also check neutrophil infiltration, and we also again observe that in a mutated status, we see that here the clear myeloid cell infiltration, but after the treating the inhibitors of NF-kappa B and JAK-STAT, we don't observe any infiltration anymore. So the last one is that, okay, that it's a cellular autonomous or not. The answer, autonomous or not, that they are regulating the plasticity is Yes, because the first that we tested is the by organoid of the mouse prostate to see after activating the PTEN, so actually we added tamoxifen to activate the PTEN deletion, but in a control data, definitely we don't see anything, and we just see that the basal lumina and then the empty lumen. But when there is a PTEN deletion, we don't see anymore. They are highly plastic, and the PTEN peak is even worse because they started making tumor already. And when we stain the Ka, the YFP, as you see again, then the P10 and P10 peak, we see the co-expression and the lumens are way smaller. However, when we treat the NF-kappa B and JAK-STAT inhibitor in this organoid, we see that we start this. So I'm sorry. So we treated those two inhibitors and then collect the cells and the seeding it for making organoid, and then we see that again compared to the PTEN deleted condition. After the inhibition, we see that they are started making the same structure as we observe in a control mouse, and their hyperplastic organoid ratio in the whole organoid is way lower. So their differences are significant. And also for sure that when we tested the staining of different markers in this organoid that compared to, so here what we did is actually do the genetic mutation. So we deleted the P65, which is a component of NF-kappa B, and then we try to see that whether those hybrid are observed or not. And the answer is definitely we don't see any hybrid after deleting P65. So this is also proving that activation of NF-kappa B is a cellular autonomous. And then when we do the organoid staining and the H-chain staining, we see that the structure is mostly same as control data. So just quickly speaking here was that we tried to see what kind of genes are activated or suppressed. So this is just confirmation of what we observed previously. So, P10, so in a P10 deleted status compared to when we delete the P65 together, definitely there is innate immunity, cytokine, neutrophil. Those all immune-related things are suppressed after P65 deletion as well, and their gene expression in the bulk RNA-seq is proving the same. So if I summarize everything dealt with in this paper is like this. So when we have a PTEN deletion in basal, definitely they will go to the tumor in the end, but there's two different paths, right? The cells are passing by. So one is passing by basal hillock and basal alumina hybrid. Proximal status going to PN and adenocarcinoma, which is faster than the basal highly expressing NK3.1, even though it goes to adenocarcinoma in the end. Here is way slower, and then they are having a totally different gene signature. So this is at the end of my presentation, and those are all the source of funding which made this work possible. And most of the people in my lab is in this photo, especially the Chen, who is a co-first author with me, Mani Lan, who was administrative support, Cedric, my co-supervisor, and Don, who is sharing my office, always cheering me up with a bunch of discussion. So thanks a lot again for listening to this talk, and I'm happy to get some questions from your side.
네, 감사합니다.
감사합니다.
질문 있으신 분들 손들기 버튼 하시고, 제가 이름을 호명하면 마이크를 켜시고 질문을 해주시면 될 것 같고요. To international student, if you have any question, turn on your microphone and ask. 네, 저도 질문이 좀 있는데, 일단 학생들이 질문이 너무
네,
많으면 시간이 너무 많이 딜레이가 돼서 일단 좀 보고 하겠습니다.
네.
네. 정현민의 학생 마이크 켜고 질문하시면 될 것 같아요.
네, 혹시 들리시나요?
네, 잘 들립니다.
아, 네. 좋은 강연 감사드립니다. 제가 질문 드리고 싶은 거는 그러면은 다른 피텐 딜리션이 있는 암종에 대해서도 이게 적용이 가능할지 궁금하고요. 그다음에 또 뭐 베이잘 타입이나 루미널 타입 같은 경우에는 브레스트 캔서에서도 있는데 혹시 브레스트 캔서에서도 적용할 수 있는 그런 부분이 있는지 궁금합니다.
일단 다른 암종은 사실은 프로스테이트 캔서를 제외하고는 피텐 딜리션이 그렇게까지 영향을 준다고는 알려져 있지는 않는데, 아무래도 이제 같은 구조를 가지고 있는 salivary gland라거나 아니면은 이제 인테스타인에서도 이 부분을 스터디를 하시는 분들이 있다고는 제가 들었어요. 근데 아직까지 결과가 발표가 되지는 않았다고 알고 있고요. 그리고 브레스트 캔서 같은 경우도 지금 제가 다른 프로젝트에서 하고 있는데 결론부터 말하면 프로스테이트 캔서에서는 피텐 딜리션이 그렇게까지 영향이 없습니다. 저희가 사실 테스트를 해봤는데 PIK3CA가 주는 이제 그 영향이 너무 크다 보니까 브레스트 캔서에서는 사실 P10이나 뭐 P53 같은 경우는 많이 이제 그 이펙트를 찾아보기 힘들다고 보시면 될 것 같아요.
감사합니다.
감사합니다.
네, 김희슬 학생 질문하시면 될 것 같습니다.
네, 강연 너무 잘 들었습니다. 제가 궁금한 건 두 가지가 있는데, 그, 그 휴먼에서도 이제 동일하게 그런, 하이브리드 상태나 트랜지션 상태가 일어나는지 확인해보신 것 같았고, 여러 튜머 종류에서도 동일하게 일어나는 걸 확인을 해주셨는데, 휴먼 혹시 컨트롤에서는 아예 안 보이는 건지도 궁금을 했고, 그리고 두 번째는 이제 마우스 오가노이드에서 이 NF 카파비타를 액티베이팅 시켰을 때 튜머가 확실히 그로하지 않는 게 보였는데 휴먼 오가노이드에서도 동일하게 나타나는지도 궁금했어요.
네, 일단 첫 번째 질문부터 말씀을 드리면 휴먼 노멀 샘플에서는 저희가 그 현상을 이제 보지는 못했고요. 마우스 같은 경우도 마찬가지였다고 보시면 될 것 같아요. 이 트랜지션이 어쨌든, 그러니까 사람이 이제 휴먼 상태, 그 휴먼이나 마우스가 완전 어덜트 상태에서는 이제 더 이상 그러니까 물티포턴시를 갖지 않기 때문에 이게 컨트롤에서 만약 그 물티포턴시가 발견됐다고 하면 다른 뮤테이션에 의해서 이게 푸시가 들어갔을 확률이 좀 있다고 저희는 생각을 많이 하고요. 그리고 마우스 오르가노이드에서 NF kappa B 액티베이션을 저희가 확인을 한 건 맞는데 휴먼 오르가노이드는 결론부터 말하면 구하기가 매우 힘듭니다. 왜냐면 휴먼 사실 제가 발표를 여기서 하진 않았지만 하나 짧게 보여드리면 제가 하나 넣어놓는데 그 휴먼하고 어덜트 마우스에 지금 보시는 것처럼 사실은 이 구조가 많이 다르거든요. 그래서 이거를 어느 부분을 떼서 어떻게 연구를 하냐에 따라서 많이 달라지기 때문에 휴먼 오가노이드 단위에서는 아직까지 실험이 되지 않는 걸로 저는 알고 있습니다. 그리고 이제 이 도너로 오시는 분들이 그 노말 샘플하고 같이 바이옵시를 해준다고 해도 이게 어쨌든 튜머에 가까운 부분이다 보니까 완전히 컨트롤이다 뭐 완전히 그렇다라고 하기는 조금 어렵다고 보시면 될 것 같아요.
감사합니다.
네, 다음에 샤피라 맞나? 네, your turn.
Okay, so in the context of P10 deletion leading to BC plasticity, how do you interpret the enrichment of AP1 family motif like the false AP1 and their potential cooperation with the signaling in activating interferon and TNF pathways? Thank you.
Okay. So actually, AP1 is more the signal of cell stress, let's say, because I mean, not only in the prostate, but in the skin or mammary gland or even dermis, for example, that if we are giving some certain stress, the very first thing happening is that AP1 is activated. Which means that the P10 deletion is sort of stress to the cells, which is coming from external, since we need to induce the mutation. So it's not necessarily related to the interferon or innate immunity related, but this means that cells are stressed enough to go further. But definitely, if we only observe the AP1, then which means those cells are just stressed, maybe going to be dead, rather than really we see something. So that's how we explain the enrichment of AP1.
All right, thank you.
Thank you.
학생들 중에 질문 있으신 분 없으실까요? 그러면 저는 캔서를 한 사람이 아니어서 잘 모르긴 하는데 저도 너무 재밌게 들었는데 그 피텐 딜리션이 NF-kappa비 시그널링을 이렇게 업레귤레이션 다이렉트도 할 수 있지만 사실 인비보 환경에서 가면 박사님 사실은 주변에
네,
있는 미세 환경, 예를 들어서 뭐 마크로파지나 이런 이면셀이나 스트로마나 이런
네,
애들에 의해서 이게 그런 애들과의 크로스톡에 의해서 이게 업레귤레이션 될 가능성은 없나요? 이쪽으로 보신 적이 있는지 이게 좀 궁금하고, 그
어,
두
네,
번째는 사실은 라파마이신을 트리트하게 되면 걔가 다이렉트로 NF-kappa비의 그를 억제한다고 생각하시나요? 아니면 사실은 라파마이신은 사실은 엠토를 이니비션 하게 되면 그 다운스트림의 뭐 트랜슬레이션이나 전반적인 셀 사이클 이런 게 다 다운되니까 그로
음,
인해서 튜머가 억제될 수도 있다고 저는 생각이 드는데 사실 이 점에 대해서 어떻게 생각하시는지도 좀 궁금합니다.
어. 일단 라퍼마이신 먼저 얘기를 해드리면 저희도 후자라고 생각을 하는 게, 사실은 제가 여기서 보여드리진 않았지만, 저희도 NF-kappa, 제 동료가 이제 그 코퍼스인 친구가 NF-kappa 다이렉트로 이제 영향을 받냐 안 받냐를 봤었는데요. 그렇게까지 다이나믹하지는 않고요. 사실 이게 좀 다운스트림이 더 있는 것 같아요. 그래서 저희가 볼 때 라파마이신이 그렇게 이제 셀 사이클이나 안지오제네시스를 이제 먼저 이제 영향을 미쳐놓고 그 다음에 NF-kappa인 것 같다라고는 저희는
음,
생각을
음,
하는데 사실 이 부분은 아마 지금 코퍼스트 했던 친구가 하고 있는 새 프로젝트에서
음,
이걸 하지 않을까라고 저희는 예상을 하고 있고 그리고 피텐 딜리션이 NF-kappa 인비보 사실 저희도 이거는 리뷰어들이 질문을 많이 줬던 부분인데 저희가 이 크로스톡을 보지는 못했던 이유가 YFP하고 앱캠하고 해서 이제 셀을 소팅을 먼저 해서 이제 시퀀싱을 하다 보니까 그 부분을 이제 제외하고 정말 이게 셀 오토노무스인지 아니면 주변에 영향을 받는지를 이제 엔서를 하기는 좀 힘들어져서 사실 그거 뭐 100% 아니라고 말하기는 어려울 거예요. 왜냐면 이미 저희가 뉴트로필 뭐 마일로이드 인필트레이션을 본 이상 당연히 스트로마의 크로스톡은 있다고 저는 생각을 하고요. 그래서 아마 지금 하고 있는 그 후속 프로젝트에서 아마 그 여력이 된다면 스트로말도 같이 보지
음,
않을까 좀
음.
그렇게는 계획을 하고 있습니다.
네, 감사합니다. 김우석
감사합니다.
교수님,
네, 저는 작은 질문 하나 있는데요. 테크니컬한 이슈인데요. 그 이슈가 아니죠. 테크니컬한 질문인데, 그 컨트롤 마우스랑 그 핏앤 딜리션한 마우스 싱글셀 유맥 있잖아요.
네.
네, 보통 저 입놀로지 하는 사람인데 입놀 쪽에는 이제 같은 마우스나 다른 마우스 다른 사람 같은 경우는 유맥 같이 그려가지고 거기서
아,
이제
네네.
쪼개서 이렇게 같이 그려서 이제
아,
셀
네.
이렇게
맞아요. 맞아요. 네.
쪼개서 하는데 지금 보니까 완전 유맥 그림이 완전 다르더라고요. 같은 셈인데 불구하고
아,
이게
네.
이렇게 하는 건가요?
사실 저희가 인테그레이션을 여러 가지 시도했다 실패를 했고요. 근데
네.
그게 어떻게 보면 좀 당연한 얘기인 게 인테그레이션을 하려면 이제 이제 같은 셀 타입을 규정하는 앵커들이 있어야 하지 않습니까? 근데 문제는 여기서 이미 나온 프록시말이랑 요 프록시말이 사실은 같은 셀이 아니고 여기 있는 이제 P63 로우와 여기 P63 로우도 완전히 같은 셀은 아닌 상황이라고 생각을 하시면 돼요. 그래서 이제 더 이상 컨트롤 없는 셀도 있다 보니까 이게 인테그레이션이 되지 않아서 완전 다르게 그린 거고 그러니까 더 이상 저희는 이제 인테그레이션을 진행을 하지는 않았습니다. 그래서
피텐 마우스 저기 이쪽 조직이나 셀
네,
피텐 마우스에서 저기 예를 들어
네.
BCP63 로우랑 저 셀도 대부분 정상인 셀인 거죠. 그러니까 피텐 마우스.
그쵸. 그러니까 여기서는 그쵸. 네, 아까 정상이라고 하기는 조금 어려울 것 같은 게 이게 저희가 셀을 어떻게 얻었냐면 피텐 이 마우스 모델에서 이게 아니구나. 마우스 모델에서 요거를 이제 딜리션을 시킨 다음에 6주간 이제 인덕션을 시켜 놓고요. 그 다음에 이제 셀을 소팅을 해서 싱글 셀을 하다 보니까 사실 요 로우 셀과 요 로우 셀이 완전히 같다고 하기는 좀 어려울 것 같습니다. 그 뮤테이션까지 고려하면.
발현이 많이 바뀌어 있겠네요. 그러니까 좀.
네, 네. 왜냐면 이제 여기서 보시다시피 여기 이제 겹쳐 있는 셀 같은 경우는 저희가 P60 로우라고 명명을 하지만 실제로 스테이닝을 해보면은 이 중간 어딘가랑 가깝다고 이제 보이거든요. 그래서 저희가 이게 완전 따로 그려지는 게 사실은 당연한 게 여기서는 이제 이 베이잘과 루미나는 완전 다른 컴파트먼트니까 여기서 이렇게 나오는 게 예상이 되지만 피튼이 딜리션이 되고 나서부터는 이 하이브리드 스테이터스가 나오고 이게 다 연결이 되어서 인피델리티가 생기기 때문에 이렇게 되고 같은 셀 타입으로 이제 아노테이션을 했지만 실제로는 완전히 같다고 보기는 어렵다라고 이제 이해를 하시면 될 것 같아요.
네, 한 가지 추가
네,
질문. 아니,
네,
마지막 사진에서 박사님 못 찾았어요. 박사님 어디 계세요? 마지막
네,
딱
마지막 사진이요?
네, 사람이
여기 있습니다. 사람이 너무 많아서
네,
찾기가 어렵습니다. 네.
보이시군요. 네. 네.
네, 이것도 전부가 아니라는 게, 네.
아, 진짜요?
와,
한
네,
랩인 거죠? 랩이 하나인 거죠?
네, 하나입니다. 네.
엄청 크네요.
사람이
네.
너무 많습니다.
네, 저는 질문 끝났습니다.
네, 박유민 학생 질문해 주세요.
네, 좋은 강연 감사드립니다. NFKB 경로 억제제를 사용함으로써 면역 억제에 대한 부작용이 있을 수 있다고 생각되는데, 전신적으로 작용한다면? 표적 특이적으로 작용한다거나 이러한 방법이 연구되어 있나요? 그리고 억제제의 장기적인 면역학적 영향을 어떻게 평가할 수 있을까요?
어, 부작용은 사실 저희가 NF-kappa 이니비션보다는 IL-1 아나킬라에서 더 많이 당연히 이제 봤고요. 그 그러니까 부작용 중에 가장 저희가 흔하게 봤던 거는 마우스 그 구강 내가 완전히 부푼 상태로 마우스가 이제 죽어 있는 경우가 많았어서 어, 그 부분에 대해선 나중에 이제 이 NF-kappa 이니비션을 이미 하셨던 분들 여쭤보니까 알러지 반응이 있는 마우스가 있다고 하더라고요. 그래서 그거고 표적 특이적 작용은 저희가 거기까지는 사실 고려를 하진 않았었고요. 근데 아마도 해야 하지 않을까라고는 이제 얘기를 했었고 장기적으로 이거를 치면 당연히 좋진 않겠죠. 그래서 이게 사실은 NF-kappa 이니비션도 사람한테 이렇게 장기적으로 하는 치료는 아직 아니라고 생각을 하시면 돼요. 그래서 그냥 단기로 보셔야 되고, 왜냐면 아직 장기적으로 이제 면역학적인 영향이라던가 아니면 사람에서의 부작용까지 알려져 있지 않기 때문에, 그 부분에 대해서는, 좀 더 연구가 진행이 되어야 될 것 같다는 게 이제 제 의견입니다.
네, 답변 감사드립니다.
감사합니다.
네, 송유라 박사님 긴 시간 동안 강연 재밌게 잘 해주시고 질문도 답변 잘 해주셔서 감사합니다. 디펜스 잘 마무리하셔갖고,
네,
네,
감사합니다.
박사학위 잘 받으시기를 기원
네,
드리겠습니다. 네, 그러면 오늘 수업 여기서 마치도록 하겠습니다. 다 모두 수고하셨습니다.
감사합니다.
네, 감사합니다.